Welcome to Bioloom Islands. Bioloom Islands is a learning space built for the next generation of biologists. A place where molecular biology, computation, and curiosity intersect. Biology is evolving faster than traditional education can adapt. Bioloom Islands is a learning space built for the next generation of biologists. Synthetic biology, place where molecular biology, computation, many learners never get the chance to use the intersect. Bioloom focuses on practical understanding faster than traditional education. We turn complex tools into a learning space built through hands-on environment, guided exploration, and real scientific biology. Place where molecular biology is one island, yet many learners never get the chance to use the intersect. Bioloom focuses on practical understanding faster than traditional education. We turn complex tools into a learning space built through hands-on environment, guided exploration, and real scientific biology. Place where molecular biology is one island, yet many learners never get the chance to use the intersect. Bioloom focuses on practical understanding faster than traditional education. We turn complex tools into a learning space built through hands-on environment, guided exploration, and real scientific biology. Place where molecular biology is one island, yet many learners never get the chance to use the intersect. Bioloom focuses on practical understanding faster than traditional education. We turn complex tools into a learning space built through hands-on environment, guided exploration, and real scientific biology. Place where molecular biology is one island, yet many learners never get the chance to use the intersect. Welcome to Bioloom Islands. Bioloom Islands is a learning space built for the next generation of biologists. A place where molecular biology, computation, and curiosity intersect. Biology is evolving faster than traditional education can adapt. We introduce the new era of human and environmental evolution. Synthetic biology. Yet many learners never get the chance to use them meaningfully. Bioloom focuses on practical understanding. We turn complex tools into usable skills through hands-on workshops, guided exploration, and real scientific workflows. Today's workshop is one island in that larger journey.
A very good evening, everyone, and a warm welcome to all of you joining us today from different parts of the world. We are truly delighted to have you, such an enthusiastic audience gathered here for this exciting session on one of the most revolutionary technologies in modern biology, CRISPR. So before we proceed with today's program, let me take a moment to introduce Bioloom Islands in more details. Bioloom Island is a pioneering synthetic biology-focused biotechnology company dedicated to advancing innovation across multiple scientific domains. Our work primarily focuses on environmental, agricultural, and biomedical research, where we design and implement cutting-edge biotechnology solutions to address real-world challenges. In addition to research, Bioloom Island also engages in commercial biotechnology projects, translating scientific discoveries into practical application that can benefit industries and communities. Alongside our research and commercial initiatives, one of our core missions is education and capacity building. We regularly organize academic programs, workshops, and training sessions to help students, researchers, and professionals learn emerging scientific concepts and modern technological approaches. These Programs are designed not only to provide theoretical knowledge, but also to develop practical thinking, problem-solving skills, and industry awareness. Throughout this session, an integrate approach, we, the Bioloom Island, combining research, innovation, commercialization, and education. Bioloom Island aims to create a new generation of scientists who are confident, globally competitive, and prepared to contribute to the rapidly evolving world of biotechnology. That's the significance of Bioloom Island. So now, when we talk about the future of biology, we can't ignore one critical transformation happening across the field: the integration of computational approaches into life sciences. Biology today is becoming increasingly data-driven. Technologies such as genome sequencing, molecular stimulation, protein structure prediction, and system biology generate huge amount of biological data. Without computational tools and analytical skills, it becomes nearly impossible to interpret these informations effectively. So, learning computational biology, bioinformatics, and digital research tools allow scientists to design smarter experiments, reduce cost, and accelerate discoveries, and even predict the biological outcomes before entering the laboratory. In simple terms, computational knowledge is becoming a superpower for modern biologists. And this is exactly where technologies like CRISPR become even more powerful. Because gene editing today is not just about laboratory techniques, but also about precise design, prediction, and analysis supported by computational tools. So with that, I know you are eager to join with today's session. So before we begin, let me give you a few quick housekeeping notes. Please keep your microphones muted during the sessions. You may have some questions during the session, so please include these questions in the chat so we can address them during the Q&A session. The webinar will run approximately till 10 p.m., including short transitions between the segments. So we encourage you to actively engage, take notes, have a great idea, and make the most out of this learning opportunity. So now let me have the honor to introduce our speaker to you. Today, we are extremely fortunate to learn about this transformative technology from the distinguished scientist and educator, Dr. Lalinka Herath. Dr. Lalinka is an expert in synthetic biology and genetic engineering, holding a PhD in genetic engineering from University of Colombo, an MSc in molecular genetics from Jeju National University, South Korea, and BSc in molecular biology and biochemistry from the University of Colombo. He has also gained international research exposure as a former visiting HRD PhD scholar at Deakin University, Australia, and an Erasmus Plus PhD mobility scholar at the University of Granada, Spain. He is currently serves as the CEO and Chief Scientific Officer of Bioloom Island Private Limited, where he leads innovation in synthetic biology, molecular diagnostics, and biotechnology commercialization. Dr. Herath is also a visiting lecturer in molecular biology at University of Colombo, SLIT, ANC, and KIU, contributing significantly to the higher education and research training in Sri Lanka. His current research focuses on supervising PhD-level projects in synthetic biology, including development of next-generation biosensors for early breast cancer detection using genetic logic gates and CRISPR-based technologies, as well as pioneering work on genetically engineered bacteria designed to combat multi-drug resistance through CRISPR-coupled synthetic gene circuit systems. Previously, Dr. Herath has held key academic and industry roles, including lead scientist at the Human Genetic Unit, Faculty of Medicine, University of Colombo, application specialist in next generation sequencing at Hades Life Sciences, lecturer in biotechnology at Huang University of Sri Lanka and lecturer in molecular biology at NSBM Green University. He has published over 50 international scientific papers and patents, with notable innovations such as the development of genetically engineered bacteria biosensors for detecting heavy metals in contaminated water and a biomarker-based system for identifying Korean abalone species in marine aquaculture. One of his most significant scientific milestone is the creation of Sri Lanka's first genetically engineered higher vertebrate, a transgenic zebrafish biosensor, making a historic achievement in the country's biotechnology research. So, Dr. Herat also played a key scientific role in Korean government-funded DNA fish vaccine development projects, further demonstrating his impact in international biotechnology and translational research. So, without any doubt, we are truly honored to have him with us today. And we are confident that this session will provide a valuable knowledge, inspiration, and practical insight for everyone attending here with us today. So, without a further delay, let us begin this exciting journey into the new world of gene editing and biotechnology innovation. So, Dr. Lalinka Herat, dear sir, the virtual stage is now yours.
uh, for introduction and very good evening, everyone. Thanks for joining this exciting session to another word that the nature gifted for biologists to manipulate, utilize biological systems. Okay. The tool is the CRISPR tool. I know all of you are very much excited to hear and also explore the potential of this interesting tool and what sort of application range that CRISPR tool uh, has, like as a very powerful gene editing tool initially, as all of you all have heard. But in today's session, we will explore CRISPR is not all about editing genes, but it has lot other implementations as well. Okay, let me share my screen first. Okay, the today's session titled as the CRISPR clustered regulatory interspace short palindromic repeat. So that's the long name, everybody. So it's compressed as CRISPR, so easier for us to use as a term. CRISPR is everywhere, so we can't use these long terms. So scientists, biologists decided to compress and come up with this nickname or the tiny name, the CRISPR. And specifically the session I titled as the Beyond Gene Editing. It's, as we all have heard, CRISPR is, whenever we mention CRISPR, we think it's a scissor, it's a cutter, we can chop DNA with it, right? So today we will explore, is it only about gene editing or chopping, cutting DNA, but the expanding universe of CRISPR technologies. CRISPR catalog is getting bulkier and bulkier, guys. So that's what we are going to explore in today's session. Right. Okay, here's a small outline for today's session. So I'll start exploring this tool, starting with the CRISPR revolution, how the revolution actually started initially, and then we will briefly go through the editing power, the DNA gene editing power of CRISPR tool from editing, and then how it actually transformed or slowly or shifted from editing aspect to some other health-wise, environmentally, or in other biological application-wise important aspects started transforming from editing aspect to the sensing aspects as well. So we will explore what are the sensing aspect of CRISPR as a tool. You can of course use CRISPR as a sensing tool. Some environmental pollutants or certain biomarkers in cancers and any other diseases. We can also use CRISPR as a sensing tool. So we are going to explore that aspect as well. And also CRISPR as a biological circuit. What do you mean by biological circuits? You know, circuits mean some programmable component. In electronics, we use the term circuit that you can provide commands and perform logics on a certain device. Similarly, CRISPR can be used as an analog to perform logics in a biological system or inside of tiny cells to provide commands and get some work done out of a cell, which is biologically significant. So CRISPR as a biological circuits, we are going to see how you can also use it, right? Another interesting aspect of CRISPR and then how it can be emerged into future uh, applications, implementation. One of the very interesting tool I'm going to introduce today, which is Tiger, guys, TIGR. It's a novel approach. Not much people in Sri Lanka, based in Colombo, talk about this tool. Tiger is a new tool that I'm going to introduce in my session today for you all and let you know how powerful Tiger is emerging or the futuristic version of CRISPR tool and how you can later use it in your research, upcoming experiments. So that we will explore in today's session. And briefly, how to design a CRISPR target. The precision engineering can be done with CRISPR tools. So how you can basically design a CRISPR target technically, biologically, or mechanically. So that aspect we can briefly touch in our session. Also, the CRISPR, as AI is everywhere now, we can also see how AI can be improved or the enhanced the capacity or the precision, gene editing power, the sensing power of CRISPR tools. Again, the real world impact, the transformative power of CRISPR, because CRISPR, of course, as I keep repeating, this is one of the most powerful weapon or a tool that biologist has ever received. If we do not utilize it wisely, we can bring about a disaster. If you know how to technically manipulate this tool, you will be the most powerful human being on the planet. I would say, as a scientist or a biologist, you can do amazing things with CRISPR tool if you use it responsibly and wisely, guys. Right. The getting started workflow for CRISPR implementation, how you can actually initiate the CRISPR project in your laboratory. So what steps to be followed that we can towards our latter session of this program, we can actually get an idea about how you can list out all your workflow in a CRISPR laboratory experiment. Yes, the future horizons we can finally discuss. So this is the outline. This is how I have organized my content today because I wanted you all to get an idea how broad CRISPR is. CRISPR is not about just, it's not just about cutting DNA, chopping DNA and pasting DNA. It has lot other implementation when it comes to biology, guys. Right. Right. Okay. 1 second. Right. So to learn, hear about this story, the CRISPR story goes back to the 1970s, everybody. So it's not that CRISPR was like suddenly invented. So it was, there's this need, there's this requirement among scientists in working with DNAs, they initially, back in the days, scientists, when you think about DNA as an information stored in molecules in biological systems. In my other sessions also, I explain how you look at a DNA as the IT unit of a biological system, because that's where all the information are being stored about biological systems in certain language, guys. So basically it's the IT unit of biological systems that stores biologically sounds information about organisms. Let's say when it comes to human, our hair color, our skin color, our eye color, our height, the way I behave, way I talk, those things are being stored in a certain language in DNA. So scientists in genetics they actually found tools to read this information. Most of you all know the sequencing technologies actually enabled scientists to read the information in DNA. So as humanity, as scientists, we didn't want to stop there. We know what information are there in DNA. But what if we can actually change the information? Right? What happens if you introduce new information into DNA? That's when actually the genetic engineering era emerged as say, because we know as humanity we want to change things, we want to experiment with things, so we are trying to change things and see what happens. And we, if information can be changed, whether these traits of organisms can also be changed. So that aspect, that's burden, that burning question was there, whether we can change this information in DNA. So if you go to the prehistoric era of CRISPR, before CRISPR, what actually happens among scientists? Right, genome editing before CRISPR. So that happens. There were few tools I want to mention which are not that popular, but they actually laid the foundation for CRISPR as a very powerful tool to come to the guys. For come to the stage, it actually laid the foundation. So if we explore that aspect, so we initially found as scientists that in a biological systems or bacteria-like organisms, there are natural enzymes that can cut DNA at specific sites and separate DNA, and then it can be merged as well with some binding material. Right. So these enzymes we had before CRISPR era was restriction enzymes. Restriction enzymes could chop DNA at predetermined, like specific targeted sites, specific sites only, not like everywhere in DNA. Right. So there were special sequences identified by these restriction enzymes that we call restriction sites, and it's chopped by these restriction enzymes. So then two more tools were discovered. Namely, one is ZFN, zinc finger nucleases. And after that, another component, molecular machinery called talent. So these two tools actually gave some hopes for scientists in gene editing, changing information, who were interested in changing information. It gave some green light that, of course, these tools showcase the potential of changing information in targeted sites, the zinc finger nucleases, and also talent as a tool. Then it laid the foundation for CRISPR to be be presented as a gene editing tool. Let's therefore, I don't want to forget these two tools, because we should always respect what had before something become popular. Then only we can understand what would come next as well. Because if something new comes, if tiger-like tools comes, you should know the history of CRISPR to wisely utilize tiger tool in future, right? Right. In 1970s, as I mentioned, the restriction enzymes discovered, like in as a certain molecule that you find them in bacteria cells, guys, as part of bacteria's defense system. DNA, these restriction enzymes cleave DNA specific short sequences, as I mentioned, that is to protect bacteria from phage infection. Again, I want to say, to understand this story, walk through this story, the science is all about discovering things, guys. There are scientists discover very interesting mechanisms which are inspired by nature, and then we see how we can actually adapt them as technologies to utilize in biological experiments, or to get use out of it as something provide certain benefits for humanity. So we learn things in biological system. We found this enzyme, restriction enzyme in bacterial cells, and then we studied what happens there, and then it was developed as a tool to cleave DNA at certain areas. So because of restriction enzymes ability, the powerful ability to cleave it, the recombinant DNA technology was enabled, guys. The unlocked cloning and recombinant DNA technologies, which means you can cut and paste DNA sequence from one place to another place in the genome or across chromosomes, guys. Recombinant DNA technology uses ligation of fragments and plasmid construct. As if most of you all are biological science background student in biomedical sciences, biotechnology programs, molecular biology programs, you learn this topic, the recombinant DNA technology, where you learn how you can clone plasmid DNA. I don't want to explain all these technical terms. Later, if you are interested, you all can reach out to me to learn properly recombinant DNA technology as well, right? Then what happens? You design recombinant DNA technology era was evolved based on the founding of the restriction enzyme. After that, what happened? The fixed recognition motif made targeting inflexible, which means editing precise genome loci in this era was still impractical because there were a lot of limitation in restriction enzyme that it can't chop at targeted site. Okay. These two tools I mentioned before, so keep them in mind, guys. The zinc finger nucleases and also the talent tool. So we can now briefly go to talent tool as well. Zinc finger protein is an interesting molecule, guys. It's a protein molecule that looks like a zinc finger-like structure is there. It can dock on a DNA strand and bound onto the DNA strand and locate certain area on the DNA with this specific structure of zinc finger, right? And then using nucleus activity, it can cleave DNA at a specific site and introduce NIC into DNA. It can later be repaired by damage repair mechanism. So what are the strengths of this? So the benefits, or what are the, when you compare it with restriction in science, what are the strengths of zinc finger nucleases? So zinc finger nucleases actually demonstrated that custom protein-based recognition, protein can actually recognize multiple areas on DNA. Recognition could direct endogenous DNA repair pathways, right? So it's very interesting. Now we have much more powerful tool than restriction enzyme. If you can actually manipulate these two arms of the DNA finger-like structure, you can protein amino acid sequence can be modified and design zinc fingers to dock on certain areas of DNA sequence. So that ability gave a certain up to certain level for scientists got the power of targeting certain areas of DNA. But what are practical challenges with zinc finger? This is a complex protein design. We have to follow complex protein design workflows in order to precisely design zinc fingers to target certain areas on DNA sequences, guys. Complex protein designs, the designs unpredictable context effect between fingers, so you can't actually predict. They were not used to predict. Even there were not much computational power to predict where these can be actually located. So it's unpredictable. The editing done by zinc finger nucleases, right? And length optimizations and variable efficiency required rapid adaptation. These protocols were not robust, guys, because it takes a lot of time to come up with a precise zinc finger design. So that was the main limitation with zinc finger nucleases. Moving on to Talon as another improved version of these gene editing tools, still we are in pre-CRISPR era, guys. Keep that in mind. So we will enter into CRISPR era soon, right? So Talon is the very close, much closer era to the CRISPR era. So this is a modular and more flexible genome editor. So it gave a lot more hopes for scientists to target genomes and then make changes at specific areas on the genome. The design principle in Talon, actually I remember our teachers were teaching Talon because CRISPR is very new. It was started emerging in 2012. I remember our teachers were teaching how powerful Talon as a tool. So I still remember that aspect. The Talent gave a lot of hopes before CRISPR era, guys. Talent used a specific sequence called tail repeats. So this is the technical aspect that I don't want to touch in this session, guys. There's a lot of technical aspect. If you want actually to learn them, you have to go through all the in-depth mechanisms, designing principles, even in zinc finger designing, and also when it comes to talent design, tool designing. So to something we will quickly go through for you all to get some directional ideas. Tail repeats. There are repeat sequences recognize a single base fusion of one. There's area course making sequence assembly more predictable than zinc finger. This is the problem with zinc finger nucleases was it was not predictable. We don't know what it's going to be doing, where it's going to be edited, even if we properly engineer the protein arms in zinc finger. Here it gives much more predictability. And also even its time, previous one was also time consuming. Advantages of a zinc finger nucleases. Again, easier modular construction. The modular construction is one of the main theories in genetic engineering and also synthetic biology. The modularity matters. So if you actually learn synthetic biology, genetic engineering topics, you know how important the modularity in precise editing, precise biological system manipulations, guys. So this gave much more easier modular construction, higher targeting accuracy in many contexts, and improved ability to target diverse sequence across genome compared to zinc finger. Still, we have limitations. So we keep exploring. We don't have the actual real tool, real weapon that we were seeking for. So remaining limitations here for talent as well, guys. Despite improved specificity, talent still required cloning each protein pair, because to come up with those tail repeats, the cloning is a much more heavy cloning protocols need to be applied in designing talent sequences to target certain areas. Cloning each protein pair, you have to clone proteins pairs separately in order to come up with a precise talent tool. Labor intensive, takes a lot of time, less scalable. For higher throughput or multiplexing, again we can talk about multiplexing as well. Multiplexing in the sense to perform multiple gene editing at a single time on different areas in the genome. So that matters when it comes to current day genome editing implementations, guys. Multiplexing is important, but that was a limitation with talent-like tools. But at a one time, you can edit only one area, still it time-consuming and labor-intensive with talent, right? As I mentioned, we can't still forget about these two tools. They gave a lot of hopes for scientists for back before CRISPR to make changes in DNA, and it laid the foundation for the conversation about CRISPR-like tool, right? Restriction enzymes, zinc finger nucleases, Talon's proved targeted gene cleavage plus cellular repair. You can merge targeted gene editing and other, you can employ the what you call the cellular repair mechanisms in order to come up with change DNA segments on a genome guide, right? Could produce precise genetic changes, and essential conceptual mindset was set up based on the foundation of the hopes given by these two tools, right? So there are technical limits, design complexity, cost and scalability, as we discuss one by one, guys, created the demand for simple. Now the demand is still there, right? Demands for simpler, more accessible tools like directly motivating CRISPR research. So here's where the story begins. The star comes out in gene editing, which is CRISPR, right? These technologies seeded methods. Why we can't forget talent and sin, right? Method, assays, and ethical framework started creating among editing changing DNA sequences with the implementation of uh, zinc finger editing, and also along with talent era, right? So the scientific community started raising their concerns about changing information in DNA sequences. It was paving the way for CRISPR to come and land on a very solid platform with these two tools emerge eras towards these two tools, guys. Right. Ethical frameworks were being developed, and still we are using the same ethical frameworks that were applied for zinc finger nucleases and talent gene editing era, right? They are scaffold on which RNA guided editing. RNA guided editing is what we are going to discuss here today, which is CRISPR, right? Editing build a global revolution. Okay, everybody. So, very interesting part. The CRISPR revolution starts. Core mechanism of CRISPR, and you can compare it with Talon and also zinc finger nucleases as well. So, what interesting here is that the CRISPR is a cutter. There are two components in CRISPR. One part is the Cas protein, which act as a nucleus and cleave DNA at specific site. And the most interesting part here is that it can be dragged by a GPS guider. What is this GPS guider towards the specific location? On it can be pulled with a GPS guider. It is like the latest BYD vehicles that we have embedded GPS system in it, right? So you have your big screen, you can go anywhere you want. Back in the day, we had all cars, we don't know where we are going. Now here we have the more sophisticated GPS system for these nucleuses to be guided towards a DNA sequence and then land on exact location where the editing need to be done. That's done by a very interesting component, which is the guide RNA component. An RNA molecule can attach to certain area of DNA, bind with CRISPR molecule, and it can be guided toward specific location on DNA where the editing needs to be done. Then again, the implementation of DNA repair mechanisms can come and heal the edited nicked area on DNA, right? So who discovered this? So we should give the respect for the people who actually discovered these tools. Discovery was done in 2012 the CRISPR tool by a great scientist called Prof. Jennifer and colleagues, and later they received Nobel Prize for this invention. As they actually discovered this interesting uh, DNA, the editing tool, while as an accident, while they were studying the bacterial immune system and analyzing bacteria's genome, they saw the presence of some interesting palindromic sequences here and there in bacteria genome. So she was curious about these palindromic sequences scattered in bacteria genome, and what purposes these palindromic sequences. Palindromic means that type of DNA sequence read in both direction the same letters, ATG, GAT. Likewise, it's readable in both directions. So you learn them in recombinant DNA technology topics. What are palindromic sequences? Okay. So very old topics. So I think you guys know those things. So I don't want to explain. So right, she anyway observed these palindromic sequences along the bacterial genome, and then realized there's something interesting about these embedded palindromic sequence. Uh, then, with further studies and experiments, she realized it's part of that weird sequences were achieved by viral DNA. And then it has something to do with bacterial immune system, defending bacteria against viruses. It can identify naturally, viral sequences can be identified by these sequences and then cleave off, recruiting Cas sequence to cleave this viral genome as part of bacteria's defense system. Right. So based on this interesting GPS guided gene editing tool, she actually he introduced entire new era. She laid foundation for an entire new era where the programmable editing tool can be utilized in precise gene editing or DNA editing. So giving huge respect to her findings, guys. So we can hear from her words what CRISPR means to the world and for us, guys, and then we can continue the rest.
I like to describe CRISPR as an editor for the genome. It's a way of changing the code of life in a precise fashion, very much like we might edit a document. With CRISPR, we can literally alter a single letter in DNA all the way up to a large segment of that text. And that means that we can now correct disease-causing mutations and help address climate change. Some of the ethical considerations around CRISPR focus on human health, especially altering human embryos. And the reason that is such a profound use of the technology is that it results in changes that are made that are inheritable. And so we have to really be thoughtful about how we would deploy a technology for that purpose. The other area where there's a lot of talk is using CRISPR for environmental applications, meaning altering whole sets of organisms. I think there's an extraordinary opportunity to address challenges of disease and frankly, preventive medicine that haven't been possible in the past. On the flip side, I do worry that the use of CRISPR and other technologies could get ahead of themselves. So it's very important that scientists and technologists and engineers stay on top of this as these technologies develop. Today, we're facing so many challenges in our society, inequities, challenges to our health, challenges to our environment. And with CRISPR, we have a technology that can, in principle, address all of these things. It's a wonderful tool that we can use to manipulate ourselves and our environment in ways that will benefit the planet.
Yes, with that, with her insights, we can actually move on to the CRISPR's mechanism, how this very interesting, beautiful tool work. The CRISPR core mechanism we can actually look at. So what she initially discovered as a, so he initially proposed it as a gene editing tool. Later, other scientists saw the potential of this tool being utilized in other implementations as well, like sensing aspect and many others, how to program cells using CRISPR tool. So many things can be done when it comes to biological systems with CRISPR. So the CRISPR's core principle we can initially investigate and then later move on to what are the broad range of applications in CRISPR. Here's the tool. When you look at the CRISPR tool, it looks something like this, guys. It has a big Cas protein, big in the sense these are smaller molecules, guys, look like a mango that you see here. It's a small molecule inside of your nucleus, right? Uh, Cas9 enzyme is what actually cleaves DNA. And when you look along this molecule, there's a loop structure, so which is known as the guide RNA molecule that can actually guide the CRISPR components. Here it's shown as guide RNA, so it belongs to this, right? Can you guys see my arrow? Can you see my arrow? Yeah, nice. Right, now the guide RNA is there to guide this I'll call this mango, this big mango like protein towards the which is Cas protein towards the editing site. Here it's an open double-stranded DNA. Certain things are annotated here, guys. I would like for you all to pay your attention. Here this guide RNA has to be located on DNA sequence. We are finding some area called PAM sequences. So PAM sequences need to be found, and then this guide RNA can locate upstream the PAM sequence and recruit Cas19 enzyme to introduce certain DNA nicks, double-stranded breakage uh, using these cutter act like a nucleases. The nucleases function is to cleave DNA. Phosphodiester bonds can be cleaved, right? So it's done with this particular molecular machinery. So here's an overlook on it. And when you analyze the annotation, in typical CRISPR systems in bacteria, it has two components, guys. The CRRNA, CRISPR RNA, which is a 20 nucleotide long stretch of DNA. So this provides the base pairing sequence that recognizes the target DNA sequence with high specificity. So that's why CRISPR become very specific as well. It has a specific DNA component that can complementary base pair with your target and then recruit the Cas only on that particular location. And tracker RNA scaffold. So it forms the structural scaffolding required for Cas binding. Cas need to later bind as well to act as the enzyme of cleaving DNA and stabilize the guide complex is done by these two RNA components. But when it was developed as a technology, what happened? Something called sdRNA, small guide RNA. So this is an engineered RNA molecule by merging CRRNAs and tracker RNA together to dock the Cas protein onto that particular location. Commonly used single guide RNA merges both parts for simplified programmability and delivery to utilize it as a technology in laboratory to edit DNA sequences. Right. Guide RNA base pairs with the target like GPS lock, as I mentioned, by making complementary base pairs, and Cas will act as the component of cleaving DNA. So basically a molecular scissor, DNA scissor guiding by a GPS tracker, right? Okay, so we talk about these special sequences, the PAM recognition site which is the molecular gatekeeper. So PAM sequences are there, need to be identified by guide RNA before Cas can be recruited to cleave DNA. PAM defines where cutting can occur. So this is specifically when you extend the term PAM, it's protospace adjacent motif. So it's a trinucleotide or longer, typically trinucleotide area that the RNA merges on it, just upstream of it, and then Cas can be recruited. It's a short DNA motif next to the target of gene editing required for nucleus activation. SP-Cas9 is the typical Cas version. So PAM for that is NGG, which means N stands for any other base. It could be AGG, TGG, CGG or something. Two Gs with another base. If there's some trinucleotide span is there, a guide RNA can be recruited at that specific site and do the job of breaking DNA strands. So PAM helps Cas differentiate self from non-self DNA, facilitate local DNA unwinding. So that's another importance of PAM sequence, having PAM sequence. Right. So if you simplify stepwise what happens in CRISPR editing, initially PAM recognition, the small trinucleotide locations need to be identified scattered across the DNA target, right? PAM recognition need to be done. Then unwinding happens. The two strands need to be unwound. Then Cas can cleave at that site. Repair mechanisms are being implemented to reseal the nicks or the double-stranded cleavage introduced by Cas protein. So that's what happened in the CRISPR editing machinery, guys. Right. Why this is precise? Certain points about precision of CRISPR tool. Guide specificity is one of the most important strength of CRISPR tool when you compare it with prehistoric tools like Talon and also the zinc fingers guide. Sequence complementary of the guide RNA. Guide RNA need to come and complement its certain target site, right? Of the guide RNA directs Cas to the exact genome base pairs. PAM gating, the molecular, it's a locker for this guide RNA to come and bind. PAM motifs restrict where Cas can engage. If there are areas with no PAM sequence, Cas cannot act on that particular areas. Guide RNA cannot direct them towards areas where there's no PAM sequences. It's beneficial on the other hand, it becomes a limitation of CRISPR tool as well. But for minimize of target specificity can be enhanced because there's PAM location. PAM sequence should be there in order to edit. Increasing target fidelity, how reliable the edit is, and preventing self-cutting. Right, cleavage outcomes can make double-stranded DNA or single-stranded nicks, cell repair mechanisms, the typical repair mechanism, neighbor joining method, homology recombination methods can then be employed in healing the repairing the breakage once the modification is done. Right, so this is a summary on precision of CRISPR tool when you compare with previous tools, guys. Right, still we discuss CRISPR as a cutting tool, cleaving DNA tool. Right, CRISPR is not just a single molecule, guys. I mean, whenever you call CRISPR, we know CRISPR-Cas9. Is that a single variant? So another question, it's not as simple as that, guys. CRISPR has so many other variants. Let's say CRISPR is like a gun. There are guns as different types, right? So small pistols and machine guns, and you have different versions of guns, right? Likewise, CRISPR has variants when it comes to gene editing as well. So let's explore this variant list of CRISPR. CRISPR-Cas, the genome editing pioneer, and initially the Cas9, SP-Cas9, Staphylococcus pyogenes Cas9 was found or discovered as the first CRISPR tool it was done by Prof. Jennifer. That's the first CRISPR-Cas system to be recognized. And by analyzing further, we knew that there's a lot of other variants of CRISPR can also be there along the genome, and by getting inspiration from other variants, we can different versions of CRISPR with different uh, strengths and benefits when it comes to different types of applications that you need to get done in the laboratory, guys. So it uses when you talk about this typical system, a guide RNA to locate target DNA and nucleus domain to create double-stranded break. It requires the PAM sequence, right? And editing was revolutionized due to simplicity. Now comes limitation. Off-target activities can also be there, because if there are similar PAM sequences on other areas, CRISPR can, there's possibility for Cas system to be located on other areas as well. Even if comparative to previous tool, this is much more minimized, there could be still there's risk for it to be introduce off-targets modification, right? Here's the CRISPR Cas9 variant family. Can you see this classification, guys? CRISPR, if you are to design a CRISPR tool in the laboratory and use that in one of your research or anything, think about what variant you are going to utilize, because different variants has different implementations, guys. It's not just like a small system. It's much more complicated than that. You have to look into what actual variant to be used as well. So it's an expanding toolbox, because scientists are keep exploring the looking at the genome of bacteria and keep mining new and new CRISPR versions like Cas versions, and then later start recruiting them in CRISPR workflows in the laboratory as a developed technology. Yeah, the Cas9 smaller nucleases, Staphylococcus, easier packaging into AAV vectors. The Cas need to be packaged in a DNA vector, plasmid vector, and then introduced. There are different benefits in different versions. There are enhanced versions of Cas9 variants. Like when you look at here, ESP can enhance Cas9, HF1 version, a hi-fi Cas9. So these are engineered versions to reduce off-target cleavage while preserving their cleaving ability. Not only CRISPR-9, CRISPR has other families as well. CRISPR-12 family, also known as type 5 family of CRISPR. So CRISPR-12 can actually, it's a single RNA effector that cut with staggered ends and have distinct PAMs. Their PAM sequences are different from Cas19 to locate the guide RNA and Cas system. Cas13 family is also another interesting family, which is being discovered and emerged as a technology now. So you can say the Cas9 family and Cas12, Cas13, these are the most commonly used Cas families. Cas12 and Cas13 are also called the type 5 family and type 6 family. The Cas13 is special compared to Cas12 and Cas19 because Cas13 can act on RNA targets. RNA targeting enzymes for programmable RNA knockdown, not DNA, but RNA can be targeted with Cas13 system. Specifically, you are working with RNA, consider using Cas13 in your applications. It has a lot of interesting techniques developed using, later we can discuss, guys, using Cas13 system. So it can knock down RNA and it can be utilized as diagnostic tools as well based on the RNA interference. This can be acting on RNA molecules, right? Here's the summary of CRISPR classification. Modern classification split CRISPR into class 1. Class 1 is what? There are two main classification. Class 1 has multi-protein complex of Cas protein. Type 1, type 3, type 4 belongs to class 1. So I want to mention this because later for you all, if you are to start a CRISPR project, you all to at least get an idea what actually CRISPR means. It's not about going to a website and ordering a Cas system. Lot to think before placing your order, at least on initiating your project, guys. Type 1, type 3, type 4 families, they are under class 1. In class 2, you have single protein effectors, which are most commonly used because easy to manipulate and it's a single protein Cas system. Type 2, type 5, type 6 I mentioned, those are the most prominent one belong to class 2. The Cas12 one and Cas13 version belong to 5 and 6 families. It goes in class 2. So these are the most useful one when it comes to CRISPR implementations, guys. So discoveries are going on. There are 46 subtypes right now. New members such as CRISPR-12 type 5, CRISPR-13 refined functionality maps. Let's see how those two can also be further utilized here. CRISPR tool is a DNA targeting nucleases with novel PAMs. PAM sequence is different from Cas9's PAM sequence for CRISPR-12, which is type 5. And CRISPR-13 is special. It's RNA-guided RNA targeting nuclease. So if you want to act on the translational, before the translational level, go for CRISPR-13. So because it acts on RNA molecules, right? For transient transcriptome modulation and also diagnostic tools can be developed in order to regulate gene expression, can be done with these CRISPR-13 like systems, right? What's more? There's a lot more, guys. Rare and emerging variants are also there. Type 4. So this is less common, but multi-protein complex is there with atypical DNA processing mechanisms. The DNA processing mechanisms is also entirely different when it comes to type 4. So unit targeting modularities can be done with type 4 version. So we can think of utilizing these different versions as well in our research, even though they are less commonly used. Type 7 and other newly reported subtypes, these are in the early experimental stage. You can read articles published by research articles how these are being developed, because these will be the future tools, guys, right? Mechanisms and utilities still under validation. Type 3 systems are there. So couple target recognition with cyclic oligonucleotide signaling and intrinsic regulatory layer. So with that, you can regulate the CRISPR tool as well. This is very interesting when it comes to synthetic biology, where we program lives. So we logically program lives. So this signaling regulatory aspect is very prominent when it comes to synthetic biology circuit designs, right? When you think about how you are going to practically use these tools, what are the obstacles, what are the hurdles, what sort of challenges you see, how to deliver this CRISPR system into nucleus, right? The smaller Cas proteins, Cas fairly bigger, hard to be delivered sometimes into the editing site. Therefore, smaller Cas proteins are being developed. Let's say Cas9, compact Cas12S, these are smaller version. Easy to package in a certain vector to be delivered inside of nucleus. So some students come to me and sometimes ask who are working on CRISPR projects. So my vector system is very big. I don't know how to deliver it into it. So I suggest them to explore these emerging tools where there are smaller Cas systems, guys, right? Diagnostic systems, Cas12, Cas13 are specifically, they are specialized tool. They are being emerged as specialized tools in diagnostic, disease diagnostic aspect. So not Cas9 is there to edit. Cas12 and 13 are being developed by certain companies as specialized diagnostic tools. We will look into two interesting diagnostic strategies developed by Cas12 and Cas13, right? The RNA editing ones are there. Cas13 has the power to edit RNA as well. Reversible modulation can be done. You can reverse the effect of RNA Cas13 because it acts on RNA. It won't introduce changes onto DNA, guys. So you can always reverse the effect, right? Diverse PAMs expand editable targets, engineered fidelity, the reliability variance reduce off-target risk, right? Combining CRISPR with prime editing, base editing is another aspect. You don't want to introduce nick to the DNA strands, but you can change the base itself only, right? So there are such strategies being integrated to enhance the precision of these tools, and point mutation without double-stranded breaks. Talk about the future of CRISPR. Keep discovering a lot of new variants of Cas systems, CRISPR tools. How? Because we have DNA. Alongside these editing tools, The DNA sequencing tools are also being developed, like NGS sequencing technologies, Illumina sequencing technology becomes more and more powerful, and also the Oxford nanopore system can read in real-time manner DNA. So along with that, the metagenomics as another area. Metagenomics means analyzing multiple organisms at a single time. Very interesting application, guys. So metagenomics, powerful metagenomic tools, we can get more and more idea about new CRISPR variants in different organisms. With that increasing discovery rate, we can get more and more tools for CRISPR to be improved. And it can be integrated with editing tools like synergistic effect can also be observed in the base editing plus DNA editing. Prime editing and new ways to deliver CRISPR system. You can use nanotechnology in order to deliver CRISPR systems, and CRISPR tool carriers can also be developed inside the nucleus. Immune profiling, regulatory oversight, there's a lot we can think about. The idea of this session is to understand how wide CRISPR is, not for you all to teach the CRISPR tool, guys. Look at the keywords and see how wide CRISPR as a tool. CRISPR's journey from Cas9 to Cas1213 also we can discuss. Right. Here's the area where I have interest on as well, because I use CRISPR as a sensing tool, which is the most uh, latest aspect of CRISPR utilization. CRISPR is not only for editing but for sensing, development of sensing tool. Most of my students work on CRISPR 12, CRISPR 13 systems in order to develop uh, environmental monitoring system, disease diagnostic systems, right? The CRISPR platform now detect nucleic acid, not cutting, but detecting, and proteins with very high sensitivity compared to the existing gold standard techniques. There are two tools I can discuss with you all, guys. The very interesting strategies, the Sherlock system and the detector system when it comes to utilizing CRISPR as a sensor detection system. So these are very high sensitive tools. Ultra sensitive detection, single molecule sensitivity enables early infection. You can go down to single molecular sensitivity. With Sherlock can detect like system developed over Cas12 protein and also Cas13 protein. And you can couple it with fluorescent readouts. Lateral flow strip and electrochemical outputs can also be obtained when it comes to biosensing utilizing CRISPR as a tool guide. Right. CRISPR beyond gene editing is what the sensing is what we are going to target. Scissors to sensors mean, scissors means cutting, sensing mean utilizing it as a utilizing that as a sensing tool. The field has broadened up. CRISPR systems now read and report DNA, RNA signatures rather than only cutting DNA. Real-time molecular detection can be done. Right. Right. I'll continue the rest from editing to sensing. CRISPR is the new frontier with detect and Sherlock. So we can discuss how detector works and how yes, how Sherlock system work in sensing.
Since the beginning of humanity, curiosity has driven discovery. We studied the stars. We questioned nature, built laboratories, industrialized science, digitized the world. Now we're living through the AI revolution. Each era changed how science was done. For centuries, biology lived within microscopes, petri dishes, test tubes, slow experiments, long timelines. But biology is no longer confined to the workbench. Genomes are analyzed with algorithms. Proteins are predicted before they're observed. Entire biological systems are modeled before a single experiment begins. The laboratory is no longer just physical, it's computerized.
To start the machine, one press this button here.
And that changes everything. Synthetic biology involves engineering life. It merges biology with computation, turning cells into programmable systems, moving innovation to the intersection of wet lab and code, which means traditional lab skills won't just be enough to cut it. The scientist of tomorrow must understand data, modeling, and machine learning. Because modern biology runs on information, and the next era will be led by those who can design it. So, you can stay home. You can open another tab, watch another webinar at 1.25 times speed, take notes you'll never read again. You can tell yourself you're learning. But let's be honest, scrolling through science is not the same as stepping into it. You can ask AI what environmental DNA metabarcoding is. It will give you a clean paragraph, maybe even a diagram, and that's powerful. AI is already inside labs, running simulations, analyzing sequencing data, and optimizing experiments. Algorithms now map ecosystems faster than field teams. Machine learning screens compounds before a human ever touches them.
The last 10 years was really about the science of AI, but the next 10 years is going to be the application science of AI.
But here's the line. AI can process data. It cannot build depth. It can generate answers. It cannot challenge your assumptions. And most workshops? Still teaching what was impressive 10 years ago. Repackaged slides, safe techniques, outdated comfort. If you're comfortable learning yesterday's science, stay online. But if you want to understand where the field is actually going, then you don't need convenience. You need proximity. Because tomorrow's scientist isn't just someone who runs experiments. It's someone who understands how biology, data, and computation collide. Streaming is intangible. Showing up makes it rare.
Genetic engineering could become our most potent tool for managing the planet. The wonders of the future may not be built with concrete or circuits, but with cells.
Okay, so you saw some exciting news, right? So we are excited to announce an exclusive upcoming training opportunity, a hands-on workshop in eDNA barcoding and synthetic biosensors for real-world environmental monitoring for next-generation environmental biotechnology. It will be held at BMICH with 3 expert speakers: Professor K.M. Nalin de Silva, Chair Professor of Chemistry, University of Colombo, Dr. Dileep Patiraja, Senior Lecturer, University of Colombo, and Dr. Lalinka Herath, CEO, CSO, Bioloom Islands, and Visiting Lecturer, University of Colombo. And this is a exclusive expert guided session. Only 3 seats will be available. So by participating, you will gain on not only hands-on experience, but also conceptual clarity in environmental DNA technologies, bioscience development, and modern biotechnological approaches that are expected to dominate scientific innovation in the coming decade. So, if you are interested in environmental biotechnology, molecular diagnostics, or emerging bioengineering technologies, this is rare opportunity to learn directly from learning experts in intensively high impact settings. So, early bird registration is open now, so don't be late. And now, let's welcome back Dr. Lalinka Herath to continue with part 2 of our webinar. Dear sir, over to you.
Yes, um, as Pabodhi also mentioned, we have an upcoming physical workshop on actually the meta-barcoding meta-barcoding and synthetic biosensors for environmental monitoring, guys. I want to mention it. So I'll take a minute to mention how important that aspect as well. So currently, the environmental monitoring is done by those sophisticated laboratory systems, so samples need to be carried in laboratory systems and then analyze what actually be there, which is time consuming, labor intensive, but that will not be the future or the near future in environmental monitoring, guys, which is very important to Sri Lanka as well, where we encounter a lot of challenges in post pandemic disorder analysis. So there's high potential for biotechnology implementation. That's why we propose this particular workshop as well for our students to learn about what are the emerging and real global technologies be utilized to monitor environment, metabarcoding aspect, and then synthetic biosensors using even the CRISPR technology is being used in that particular aspect as well. Guys, I invite if there are any interested students, they can also be joining our upcoming workshop, which we have intelligently planned for student to gain actual real technological skills. So that's the idea of Bioloom Islands as well, to bring about real world technologies for our students here, mainly based in Sri Lanka and our region, guys. Okay, right, back to topic, CRISPR. Let me share my screen. Okay, so this is where I stopped. Editing to sensing, the CRISPR can be utilized as a sensing tool. There are two tools I've been talking about, the detector tool and the Sherlock tool, guys. Right, what detector means and what Sherlock means. So this is what we want to understand. Sherlock is a system developed over one of the CRISPR types, which is CRISPR-Cas13 type that I mentioned. Sherlock, the long term is that specific high sensitivity enzymatic reporter. Uh, Yes, reporter unlocking. So that's what Sherlock stands for. So it was developed at the Broad Institute. So it's developed over the Cas13 tool, guys, that can be utilized to detect RNA molecules. If you want to detect RNA molecules inside of biological systems, you directly use the Sherlock system commercially developed as a sensing setup. So we will investigate how this sensing setup work based on Cas13's protein, and this is ultra sensitive compared to other sensing strategies. It can go down to the single molecule sensitivity through certain isothermal amplification strategies paired with Sherlock system, and also the CRISPR's collateral activity. And another interesting part is that you can multiplex Sherlock system, which means multiple detection can be done at a single time by utilizing Sherlock systems. And it's field friendly. What do you mean by field friendly? You can bring about the detection aspect into the on-site detection can be done with this Sherlock system. How to do that? Design for rapid, low resource workflows, very important for Sri Lankan scientists to develop low-cost detection system. You can use Sherlock strategy. So ideal for point of care use of environmental monitoring, disease diagnosing, and in Sherlock, you have to collect the sample specimen and then you have to do a bit of isothermal amplification of your target to be detected, right? And then next step is Cas13 pairs with target RNA because it works on RNA molecule if you want to specifically detect mRNA molecule. So this work on target RNA molecules, guys. Guide bind to target RNA. Readouts can be obtained by a certain signaling molecule or reporter molecule coupled to as a prop to that particular RNA molecule. There's a probe attached. So that's how the Sherlock system developed. I'll show you a diagram of Sherlock system as well. So this amplified target isothermally and then you can perform detection of presence of that molecule can be detected using Cas13 based Sherlock system. Other interesting detection system developed using CRISPR Cas protein is that which is developed over Cas12 system. It is a system developed over to detect double-stranded DNA, guys. DNA endonucleases targeted CRISPR trans reporter. That's what detectors stand for. So it targeted DNA targeted CRISPR sensing can be done by detector using Cas12. And if you want to read more, the Sherlock is specialized RNA detection molecule, right? Cas12 mechanism works like this. So it recognizes double-stranded DNA targets and it can single-strand DNA's activity can be implemented on that particular double-stranded DNA, right? So then detectable signals can be obtained. Rapid lateral flow is there. Detector translate collateral cleavage into lateral flow or fluorescent readout. So you can create sensors enabling instrument-free tests for pathogen like coronavirus detection was actually done using this particular tool. There's a tool developed for detector system as well as Sherlock system as well. So it's very much specified. It's high specificity tool developed by a certain company. Guide design and target selection afford discrimination between closely related sequence. You can actually identify the difference between closely related viruses or the DNA, double-stranded DNAs using detector system. So useful for variant and mutation calling, right? COVID-19. So if you look at what are the actual real world application of these sensing systems based on CRISPR. So as I mentioned, a platform like My Sherlock developed by one of the companies. So it combines low cost hardware and a paper strip readout. It's a paper-based detection system developed over Sherlock system and phone-based interpretation. There's a mobile app as well coupled with that to interpret the data readouts to deliver rapid SARS-CoV-2 virus and mutation, single mutation detection can be done very rapid manner in about 1 hour. So for mutation analysis, it's a significantly fast, very fast time period. Right, detects key mutations in these viruses, minimal user steps suitable for decentralized settings, smartphone integration automates analysis and data reporting can be easily done, right? So these are the two tools how they work. So for you all to get an idea of the mechanisms of these two systems, guys, look at these two diagrams. Sherlock shows how it actually works. So you go to the bottom of this diagram on the Sherlock, guys. What happens here? There's RNA molecule in pink color. The target site is indicated. We have a probe attached to this particular target site. So this probe is what this we integrated with this red color signal component and the green color glowing area. So what happens initially, it's attached to this single stranded RNA molecule. Upon the guide RNA recruited onto that area, the Cas13 can also be directed to that area. And then Cas13 can chop or cleave the probe to release this green color reporter molecule, which is a fluorescent molecule, right? So when the target site is there, Cas13 can cleave this probe to release the fluorescent signal. Basically, by analyzing fluorescent readout, now you can understand the presence of that specific RNA sequence with Sherlock system. So it's very interesting, guys. It's developed by one of these companies. Go on to the detector system. Detector system similarly act on double-stranded DNA targets. Look at this. This DNA target is there indicated in pink color. The Cas12 is recruited. The probe is released and chopped by Cas12 to release green color fluorescence readout. So this strategy can be utilized in developing specific detection of RNA molecules and specific detection of DNA molecules, guys. Right. So you can actually compare detect and Sherlock here. I have included all these information on my slides. Later you all can leisurely go through them. I don't want to, just wanted to highlight that sensing can be done with these two specific types as well, and a lot of sensitivity wise, accessibility wise, and speed wise, they are very powerful tools, guys, and we can revolutionize the theranostic, like the diagnostic field of diseases, as well as the environmental monitoring can also be revolutionized with. They are very competitive. They can replace existing chemical methods of detection, which are known as sophisticated setups in the laboratory to analyze certain environmental uh, components, and also the biological biomarkers, right? So these tools later you all can study and start researching about them. Right. So you can actually bring CRISPR diagnostic from laboratory to field based on these Sherlock and detector-like systems. With what? The enzyme engineering strategies are there to improve Cas's ability to minimize the off-target effect, right? And multiplexing is another limitation. Though by engineering these enzymes, multiplexing can also be enhanced. And then integration with digital health for remote monitoring. We can actually come up with so high throughput monitoring systems, which are like maybe you can obtain signals into your smartwatch on analyze it on your mobile phone. So that can also be done with this CRISPR based detection system developed by Cas12 and Cas13, guys. Clinic deployment will push CRISPR sensing into routine care. That's why some of our students are also working on certain cancer diagnostic tools. Breast cancer diagnostic systems are being developed even in Sri Lanka currently, which our supervision to develop breast cancer biomarker detection system to Cas13 Sherlock systems. Guys. So those are the applications we want to look at here. Right, engineered enzymes for speed and specificity, real-time multiplex panels for comprehensive screening, interoperability is another theory comes in the synthetic biology. We learn in fundamentals of synthetic biology, guys, with apps and cloud survivors. Right. Right. Here's my specialized area. The CRISPR has a biological circuits. So you can program, you can control cells in logical manner using CRISPR logic. CRISPR can be logically manipulated inside of cells and then provide commands for cells to behave in the way that we want them to behave, right? CRISPR is a toolbox for synthetic biologies. So it can be building logic gates, electronic logic gates. Similarly, we can develop genetic logic gates inside of biological system, where CRISPR can be acting as on-off switches of these logics in the small cells. And CRISPR can also be used in memory modulators, and we can use them in feedback signal loops in order to reprogram biological systems or cells. So these are one of the three core concepts in synthetic biology. We design logic gates in cells, and CRISPR can also be utilized in developing and or not like logics in cells for us to provide inputs and get some useful work done out of cells. So the architectures using CRISPR-I and CRISPR-A, two more versions, right? CRISPR interference and CRISPR activators. Those are the Cas protein has been modified in a way it can interfere the gene regulation machinery. And it, once the Cas protein is bound, it won't cleave the DNA, but CRISPR-I will block the transcriptional process, the RNA, DNA polymerase, the ability to access and create proteins, right? That's what happened with CRISPR interference. And CRISPR activators can come and bind and recruit transcription factors, which can enhance the activation of mRNA production. So you can create on-off switches, gene on-off switches using CRISPR modified versions, CRISPR-I versions and CRISPR-A versions in creating logics in synthetic biology, developing synthetic cells. Cellular memory is an interesting component when it comes to neurogenetics. So it records transient event with durable molecular marker useful for lineage tracing and biosensing. What do you mean? These are a bit advanced concept, but I'll simplify. So if you can introduce permanent changes into DNA using CRISPR, you can inherit it into another cell, which means you can cellular memory can be embedded. That information can be passed down from one cell to another cell, right? So you can program cellular memory by introducing permanent changes into DNA using CRISPR tool. Feedback control, feedback loops, which means bestable switches, dynamic regulation, stabilities, expression, enabling robust therapeutic circuit. I'll give you an example why feedback control matters. Let's say you want to develop a therapy. There's a certain disease condition, let's say a neurological disease condition. You want to target one of the genes to be down regulated. But at certain condition, the down regulation should be stopped. That happens, the CRISPR can pass information when to turn on and when to turn off if you use different version of Cas protein. So therapeutic circuits, the engineered therapies can be developed using these CRISPR programs. So even our undergraduate, one of my undergraduate also working on a therapeutic, conceptualized therapeutic gene circuit using this kind of CRISPR logic that he is trying to look at the Parkinson condition, control Parkinson condition by lodging Cas protein, and whenever required, Cas protein need to be released, right? Okay. CRISPR's biological circuits, again the same topic, programming cells with logic, memory, and feedback loops. So these theories can be, the intelligent cells can be developed using CRISPR logics. Right. Think about the programmability and machines. Cells as computers. Now cells can behave. You can brainwash your cell to behave like a computer and provide instruction into cell by combining CRISPR in a different logical manner. And you pass an input, your CRISPR molecules can turn on and off according to your input molecules. So cells can behave like computers. If you integrate CRISPR, programmable CRISPR systems into cells, CRISPR repurposes cellular machinery to compute decision. So you can basically, cells can start computing what decision to be made, and you can influence that decision by your CRISPR molecules, normally handled by electronic circuits in electronics engineering, right? Programmable guide RNAs and nuclease variants regulate gene expression with high specificity. So that's the core in controlling cells with CRISPR systems. So disease therapeutics can be developed, programmable therapies, Biocomputing is another entirely new topic. You use cellular system as behavioral. You can use biological tools for computing aspect. Biocomputing is heavily done by CRISPR variants now. And advanced diagnostic tools can be developed by coupling CRISPR with synthetic biology, logic gates and gene circuits, and also another layer to improve enhance the precision of these tools. You can think of quantum biology strategies as well, guys, along with CRISPR quantum aspects. Right. Logic gates in cells, building decision making. You can teach cells to make decisions at what actual time for a specific therapy to be activated. CRISPR systems implement fundamental logic operation inside living cells by coupling sensors to guide RNA. Circuits evaluate molecular input and produce discrete gene expression outputs. And means two inputs are given. You let two CRISPR component to activate and then to interact with the regulatory region of a gene, and only in the presence of two inputs that gene will activate and gate, or in the presence of this molecule or that molecule activate to a gene circuit, the signal is passed by a guide RNA directed Cas protein. Not, there's something called decas as well. It's a dead Cas, dead version of Cas. So this Cas can be utilized in making NOT gates. In the presence of these molecules, dead Cas goes and bind on DNA, do not activate the expression of gene if this molecule is there. With dead Cas or decas version, you can achieve that not logic inside of cells, guys. Multi-input systems can be developed. As an example, there are 4 input logics. A lot of multiple input can come and interfere with gene expression in cells based on different multiple Cas systems. So these are recent studies you can read, guys. Engineered E. coli cells can be controlled with 4 inputs. There are articles on these areas, guys. Enable condition therapies and environmental sensitive responses. Right. Cellular memory extends towards genetic record. The targeted edit write a permanent permanent history useful lineage, as I mentioned that before. So if you make changes directly on DNA, you can give permanent memory markers for DNA. Epigenetic records. Epigenome is which is not in the genome but influence the genome, like DNA methylation, histone modification, that can also be controlled by CRISPR tools, and stores transient event without altering sequence. Short-term memory can be influenced by working on epigenome, right? How these are important clinically. So it's integrated genetic and epigenetic programming that demonstrate in human T cells. This is an example. The T cell reprogramming is one of the active field in cancer therapies. The T cells can act on cancer cells in order to kill cancer cells directed by biomarkers. So we heavily use Cas system in order to obtain biomarker based signals inside of T cells in order to T cells to be reprogrammed as a therapy for targeted immune therapy towards cancers. Right. Feedback controlling, I previously mentioned as well. So these are the technical aspect. The negative feedback loop can be created. So it limits overshoot by coupling output to repressors. You can couple the repressor, gene repression system with CRISPR interference. Positive feedbacks can be developed as activations, which CRISPR A systems, CRISPR activator system. Inducible control, the Bistable switches, chemically inducible CRISPR-Cas9 circuits achieve ultra-high dynamic range, right? So there are such strategies in CRISPR. Why I showcase all these things is that how broad CRISPR is. The topic is not how to use CRISPR tool, how to edit DNA with Cas protein. That's not the topic here. Topic is how broad CRISPR is, where it evolves, where it goes, right? So that's what we are discussing, right? Beyond DNA editing, CRISPR-associated proteins also influence mRNA translation, adding an orthogonal layer, another layer of control, and richer circuit designs in synthetic biology. Look at this figure, guys. CRISPR-Cas9 is there to edit DNA. CRISPR-Cas system induces site-specific double-stranded breakages in genomic DNA. Primary system uses in current clinical studies to edit. And we, not only that, we have CRISPR-A, CRISPR-I system, as I mentioned. So they catalytically deactivate Cas9 modified to inducibility activate and represent gene expression to create gene on-off switches. The synthetic gene switches can be created. Look at this aspect. Base editing can be done. Fusion of Cas9 to nucleotide modifying enzymes. You can fuse Cas9 with another component which is nucleotide modifying enzymes. So you only modifying the base component, not the entire nucleotide. For target cytosine and adenine substitutions. So you can only substitute base. So that will not damage the DNA double strand. And prime editing. Prime editing is what Cas9 fusion with reverse transcriptase. You can fuse this Cas protein with multiple adapters for different types of editing. Reverse transcriptase that enable programmable rewriting of DNA sequence. You can actually rewrite the information in DNA sequences using prime editing. RNA editing. RNA editing, Cas enzymes, Cas13 and Cas7 to 11 family. Remember I mentioned Cas13, Cas7, 11 family are currently being developed for diagnostic and therapeutic application at RNA editing level. Transcriptional level. After transcription, translation happen to act on RNA molecules. Right. Combine transcriptional and translational control to reduce leak. Why these are important? And sharpen responses. Cas-based translation logics and cascaded are being studied. Lot of new areas to be explored, guys. Right. It's about programming a single cells. Here we discuss the cell programming with Cas systems. Not a single cells can be programmed. It's not that a single cell can be programmed with CRISPR. Multicellular computing is another interesting application when it's come to cell programming. The two or multiple cells can be programmed at a single time, or cell can communicate between each other by utilizing CRISPR programmable signals, right? You can distribute computation aspect of CRISPR into two cells. When engineered cells exchange information, guide RNAs or signaling molecules act as messages to coordinate group behavior. So you can program multiple cells or tissue to behave in a coordinated or synchronized manner by splitting the guide RNAs into multiple cells. This is called distributed logics in cell programming. It's important distributed computing tasks by splitting logic across specialized strain. Different cells strains, you can split the logic into two or more cells, right? Tissue level designing can be achieved, not a single cell level, but tissue level design. Enables pattern formation, coordinated drug delivery. If you want to target an entire tissue with a certain drug, you send the drug and let it activate in multiple cells. Send a signal, let the same drug to be activated in a multiple cell in a coordinated manner to target an entire tissue. If there's a diseased tissue, localized tissue area, right? To deliver coordinated drugs. Smart biomaterials can be to locate curves. So many applications are there when it comes to therapeutic aspects. Okay, we talk about CRISPR's current applicability. We can't forget what are the challenges we still facing. There are off-targets edits. Still, CRISPR is not the answer for everything, guys. CRISPR has a lot of off-target activities compared to previous version. It's minimal, but still it has off-target edit activities. That's why we have to utilize a lot of computational validation before you actually implement it in wet lab workflows to understand how much of off-target effects are there and whether it's permissible to be utilized in a wet lab workflow. So you want to learn these tools as well to properly conduct a CRISPR experiment. Circuit leakness need to be studied, right? Scaling complexity. It's difficult to scale up CRISPR editing. Integrate many components while preserving robustness and predictability need to be preserved as well. Translational hurdles are there. How to deliver these bulky CRISPR systems into cells and the nucleus. So some researchers who are working with CRISPR even locally and also globally, they struggle with this problem, how to get it delivered into cells. So they have this question right now. I know some of my students who are currently encountering this issue working with the CRISPR systems, guys, right? Delivering problems. Immune responses can be activated towards CRISPR systems and regulatory pathways for programmable therapy. So those are the things to be considered. But still, this is very powerful. Programmable cell therapy is living diagnostic. So very interesting. The living diagnostic are what? You can conceptualize a cell to activate their own medicine inside of the cell towards certain disease, right? Right. Adaptive biomaterials can be developed ethically. This has to be ethically done because CRISPR is not the answer for everything right now because it has off-target effects. So strictly regulated, must be strictly regulated. So this is a very powerful tool, and you have the trump, but you have to use it wisely. So that's about CRISPR's future, guys. Uh, the promise of CRISPR in biological circuits, programming cell with logic, memory, and feedback turn, uh, yeah, living systems into adaptive devices and design principle eventually. So these things we actually discussed, so I'm going to skip them, guys. Right. So here's something nobody has ever discussed here in Sri Lanka onset, that tiger tool. I keep mentioning it at certain conferences and symposiums that tiger is going to be emerge in future. So if we can get on top of it, we can actually do amazing thing as Sri Lankan science is. Tiger is the next era of CRISPR. It's known as transcriptional interference guided by RNA, right? So why this is important, tiger as a tool? So you can explore the potential of tiger. So it regulates the modification at transcriptional level without cleaving DNA. So you can integrate reversible changes to editing protocols, workflows with tiger tool. Safe to control over gene networks. You have gene networks to be controlled. With CRISPR-Cas9, you are going to edit the genes itself. But here, you are acting on transcriptional step. So you won't edit the gene, but the regulation. Non-destructive regulation reduces genotoxic risk. With Cas9 tool, there's genotoxic risk. And this is very much fine-tuned. Graded control over expression level is being done. Promising route for reversible therapies. Therapy will not be permanent. Once the disease is cured, you can reverse the therapeutic effect. If you develop it with Tiger TAS system for CRISPR-Cas, Tiger TAS, right? You can read and complex circuit designs can be achieved. Right, here's how it evolved. CRISPR in 2012, it was announced as a powerful tool, right? The limitation, large Cas9 size, more benefits are there with Tiger system, guys. Strict time requirement, here the limitation, Cas9 is larger compared to Tiger. test system, and it's strict PAM requirement. PAM sequence requirement is there with CRISPR system, but not with Tiger task. So it's another advantage with Tiger task tool. Delivery challenges and off-target risks, they are with CRISPR tools, still there. Discovery at Duke announced in 2024. So these are very new. That's why the people, some of our researchers who haven't heard about the Tiger Task tool, but we discuss those tools in real-time manner, guys, and see the potential ahead of time and see how we can utilize these tools. It's a dual guided RNA architecture with small task protein. Compared to Cas protein, task is smaller. So you can easily deliver that system into the cells, offering a different path forward. Like compared to CRISPR, this tiger will be emerged. But do not try to again rely on tiger is everything for this is the answer for everything. Tiger has some limitation as well. Always go for the synergistic aspect. Something Tiger, of course, Tiger is the look at the power of CRISPR as well, because CRISPR has been much annotated compared to Tiger, but Tiger has these areas. If you're interested in learning how CRISPR is being directed or evolved, right? So these are some benefits of Tiger test system. It's a dual guided precision system. I'm not going to illustrate as time is running fast, guys. It's a dual guided precision system, and it is PAM independence, compact. It's much more compact compared to test protein is compact compared to Cas9 protein. Uh, it's a transient mRNA editing tool. Tiger edits mRNA transiently, enabling reversible regulation with reduced risk of permanent off-target damages. Right. Tiger CRISPR, you can compare and get an idea leisurely, right? Right. So we want to understand where's the future goes. The discovery of Tiger in 2024 to in 2020, 2012, the CRISPR came. There will be more tools being discovered and unveiled in future. So we can't just stop here. We have to look, keep an eye on what's happening. Look, uh, keep scanning for more and more tools being emerging. So that's how a field evolved. The pre-chemical optimizations will happen, refine efficiency, safer dosing. Those things need to be done. Design safe, efficient delivery platform, and then it has to go to clinical trials, very important. And these tools are very powerful. It can change information, guys. So that's it's part of the parcel that when CRISPR and these gene editing come, it's a part of the package and parcel that ethical aspect comes in. So I train my student initially to think about ethics, whether we are going to do what we are going to do is ethically sound. Why we need to make these changes? It's not about whether we can, it's about whether we should do it. So that's what initially we have to think, because we are playing with uh, nature's logics, which are very much precise. So if you want to change it, you have to have a lot of justification in science, guys, if you want to change something in nature. So that's why ethics and safety aspects matters when it comes to CRISPR like tools which are more powerful. Otherwise it's like a loaded gun given to a child, right? Designing CRISPR targets, computational power is there. You want to explore the recent tools, computational tools like chop. It is one of the common tools that you want to utilize in designing your CRISPR guide RNAs to see whether there are off-target effects in your CRISPR design, guide RNA design. Chop is the kind of gold standard computational tool to design CRISPR guide RNA. So you will be utilizing heavily initially chop tool if you want to design CRISPR guide RNA. CRISPR off, Open CRISPR one, these are intelligent tools recently developed. So it streamline target selection and accelerate the iteration of these tools. AI optimization strategies are also they are now being integrated into CRISPR workflows. So models can be developed, computational mathematical model, machine learning techniques can be implemented in order to train multiplex high throughput CRISPR editing can be predicted initially before we implement it, right? With AI models. Model learns sequence to activity relationship to recommend high precision guides. Off-target reduction can be done. In silico means computational screens and genomic context scoring reduce unintended edits. It's about how much precise what we've been doing with CRISPR tool. Right, computational tools can be used, of course, as I mentioned. Precision editing, the CRISPR revolution needs AI now. Precision editing can be done with AI tools. CRISPR-Cas system use guide RNA to target genome loci for precise edits across genome. AI acceleration happens. Machine learning converts large experimental data set. The machine learning algorithms can deal with large experiments data sets and give us pre-information about how CRISPR can be. Multiplex CRISPR systems can be act simultaneously on a large genome, guys. So these tools are being utilized by recent applications. Like if I mention namely, the Colossal Biosciences recently announced that they work on they use CRISPR tool to de-extinct, bring back about direwolf by editing 14 targets across the gray wolf's genome. So to do that, they develop their own AI tools in order to predict before they actually implement it, to predict how the changes occur in across gray wolf's genome. So they have a lot of patents on these tools. They have copyrights on these tools that they developed over machine learning tools to initially predict how this would actually act. So you can explore those areas as well, how AI can be implemented in uh, enhancing the precision of our CRISPR editing workflows. Right. Design my balance high on target activity, minimal off target. So that's the challenge, guys, when it comes to CRISPR. Ask lot of questions. CRISPR is not that straightforward, even if it's a very powerful tool. Before you implement it, ask a lot of questions whether this could make different unwanted changes in other loci in genome. So those are the questions in at initial phases you know to ask. Otherwise, it's a very powerful and a dangerous tool. We have examples from our past as well. I'll let you know later. So here's a bit about deep learning aspects. Models trained on trained on uh, tens of thousands of experimental measured guide RNAs predict on target activity strong accuracy. So prediction can be done by deep learning tools. Sequence containing spam, loci nucleate can be analyzed. CRISPR-on is a deep learning tool recently trained on lot of guide RNAs. It's see the number 23,902 guide were trained. CRISPR-on tool was trained on this much of guidance. So they are very much intelligent. So you can use them as a supporting tools for your workflows, guys. Right. Here time runs out. There's lot CRISPR you can talk about on CRISPR weeks, months on it. Lot of contents to talk about CRISPR. So here's a little break on from CRISPR. Some tools I have listed down. Chop is the gold standard computational tools to uh, understand the efficiency of CRISPR tools, and off-target scoring can be done by Chop. CRISPR off tool is there, so it focuses on minimizing off-target via entire genome wide analysis is done by CRISPR off tool. Open CRISPR-1 is an intelligent tool. It's an AI-powered tool that you can develop utilizing CRISPR workflows, right? So later, I think we can share these materials with you if it's possible. I'll check with my group. So team, if it's so, you'll get the material as a guide, and then there you can read a lot. Okay, so yes, it's about AI, CRISPR guide design. Um, Here's how it evolved. Small guide for you all. Initially define the target. If you want to start a CRISPR project, what target to be, what area, specific area to be targeted. Then you design your guide RNA, which is the GPS system, right? Off target, sorry guys, off target validations can be done, and then implement and monitoring should be done. So this is not straightforward. Some students and some researchers think this is just a plug and play system. No, it's a design, test, learn cycle. That's why even the greatest, even the inventor herself sometimes thinks multiple times before implement this tool on a certain gene editing workflow, guys, because we don't know, there could be unnecessary outcomes. So we have to be precise, make predict as much as you can, then implement it and see and learn it in the laboratory before you bring it out about the field, right? How it expands to along tiger-like tools and AI integration, CRISPR can definitely be extended in the future via diagnostic, environmental, agricultural fields, because plant DNA can be edited with these tools to control the traits in plants. A lot of applications, guys. Right. Finally, yes, the CRISPR is about wide range of applications on editing. There are FDA approvals coming along for gene therapies because CRISPR is very powerful now, and there are plants being edited. Recent announcement on Cleo cleaving the Down syndrome's additional chromosome with the multiplex CRISPR system, HIV silence, and there are if you unethically conduct, you have stories from past. The human immune system could be affected if you do not responsibly use these tools. So we have to be very precise and careful, but the potential is huge for CRISPR.
What if you can design biology, not just study it? What if your ideas can turn into biosensors, gene circuits, research papers, patents, or even startups? Welcome to the Synthetic Biology Internship Program by Bioloom Islands. Bioloom Island is where biology, technology, and design come together. We believe synthetic biology is not just about experiment. It is about thinking like a designer, innovator, and a problem solver. This internship is built for students who want real skills, real outputs, and real impact. This program is open to undergraduates and graduates from biotechnology, molecular biology, biochemistry, biomedical science, bioinformatics, and related life science fields. The internship runs for 6 months, divided into 2 3-month phases, and follows a hybrid model. 4 days remote learning and 1 day of physical session per week at Bioloom hubs or partner labs. In phase 1, you will build strong foundations in synthetic biology. You will learn about biological paths, gene circuits, logic gates, and biosafety and ethics. You will design and stimulate your own mini project such as diagnostic biosensors, therapeutic gene circuits, environmental or agricultural solutions. You will also gain hands-on lab exposure, PCR workflow, molecular cloning, and DNA microinjection, and learn how to write a scientific review article using real research databases. In Phase 2, things level up. You will convert ideas into original research concepts, advanced stimulations, and publishable work. You will be trained into research paper writing, patent drafting, and intellectual property fundamentals, and even explore startup incubation and commercialization in biotechnology. By the end, you will have a strong professional portfolio, not just a certificate. This is more than an internship. It is a launchpad into research, innovation, and future biotech careers. Slots are limited. If you are ready to learn, design, stimulate, publish, and build, send your CV to careers@bioloomislands.com.
Okay, so with that, thank you very much, Dr. Lalinga, for your insightful and engaging session. We really appreciate your time and expertise. Hopefully, our audience got the maximum out of that. Okay, so now you saw a video. It was about the Bioloom Islands flagship initiative, the Synthetic Biology Internship Program. So at this moment, I would like to briefly introduce about this one of the flagship initiative by the Bioloom Islands. This is the structured 6-month hybrid internship delivered into 2 progressive 3-month phases, specifically designed for undergraduates and early career life science students in biotechnology, biomedical sciences, molecular biology, and related disciplines. The program focuses on building strong foundation in synthetic biology while gradually advancing into more specialized areas such as gene circuit design, computational stimulation, scientific writing, patent drafting, and startup orientation innovation thinking. So participants engage in guided mini projects, review and research article development, and gain exposure to industry relevant tools. So the goal of this program goes beyond conventional training. It is designed to help students think like a researcher, innovator, and a future biotech professional, developing problems, solving abilities, interdisciplinary thinking, and confidence to work in a modern scientific environment. Okay, so that's about the internship program. And at this moment, I have something exciting news to share with you. It is about CRISPR 0 Hour. So this is a game by Bioloom Islands. We are excited to introduce a short interactive learning challenge for everyone interested in genomics, CRISPR, and biotechnology. So we are launching a CRISPR 0 Hour, an educational genome analysis game organized by Bioloom Island. In this challenge, the participants will analyze 2 fictional DNA sequences, each 1,000 base pair long. The sequences are almost identical, but one contains single nucleotide mutation linked to antibiotic resistance in a stimulated scenario. Your task is to compare the sequences and identify the exact position of the mutation. So you will have only 24 hours from the moment the sequences are released to complete this challenge. Okay, so once you identify the mutations, there are several steps that you have to follow. The first thing, you have to take a screenshot highlighting the mutation nucleotide. Then you have to share it as a story in your Instagram and Facebook profiles. Not only that, but you have to mention Bioloom Islands in both stories. So I hope the 3 steps are clear. Once again, firstly, what you have to do, you have to take a screenshot of the highlighting mutated nucleotide. And you have to share it as a story in your Instagram and Facebook profile by mentioning Bioloom Islands. So the participants will be eligible for a raffle draw, but you have to follow these rules. To be eligible for the raffle draw, you have to follow Bioloom Islands through Facebook, Instagram, and LinkedIn. So all participants who correctly complete this challenge within 24-hour window, the follow by submission rules, will be entered for the raffle draw. The lucky winners will receive a fully sponsored seat at the Next Gen Environmental Biotechnology Workshop. It will be held at the BMICH on 4th of April 2026, and it will be valued 15,000 rupees. So if you are the winner, you will get that sponsored seat. So full challenge instruction and the DNA sequences and the submission guidelines will be shared immediately after this webinar to you. Then we encourage you to participate, test your analytical skill, and enjoy this hands-on learning experience. So you all are welcome for CRISPR 0 Hour. So I think all the guidelines are clear to you, what you have to do. Then we can come back to our webinar. Okay, so back to the webinar. I know you all are interested about the certificate that you can get from this webinar. So to take that, you have to complete an assignment and upload that. All the details about the deadline, submission process, and further details will be shared you through the WhatsApp community after this session. So if you want to join the WhatsApp community, our group has already shared the link for that community in the chat box. So by using that link, you can join to the WhatsApp community. Right. So with that, we will open the floor for questions. So I know at this moment also you have posted some questions into the chat box. Still, if you have some questions, feel free to ask anything related to this webinar. So Dr. Lalinka is ready to answer for your queries. So don't hesitate. Your questions may help for others as well. So you can turn on your mic and ask the question, or else you can put that into the chat box. So you all are welcome to ask questions. So let's begin the Q&A session.
Since the beginning of humanity, curiosity has driven discovery. We studied the stars. We questioned nature. Stories, industrialized science, digitized the world. Now we're living through the AI revolution. Each era changed how science was done. For centuries, biology lived within microscopes, petri dishes, test tubes, slow experiments, long timelines. But biology is no longer confined to the workbench. Genomes are analyzed with algorithms. Proteins are predicted before they're observed. Entire biological systems are modeled before a single experiment begins. The laboratory is no longer just physical, it's computerized.
To start the machine, one press this button here.
And that changes everything. Synthetic biology involves engineering life. It merges biology with computation, turning cells into programmable systems, moving innovation to the intersection of wet lab and code, which means traditional lab skills won't just be enough to cut it. The scientist of tomorrow must understand data, modeling, and machine learning. Because modern biology runs on information, and the next era will be led by those who can design it. So, you can stay home. You can open another tab, watch another webinar at 1.25 times speed, take notes you'll never read again. You can tell yourself you're learning. But let's be honest, scrolling through science is not the same as stepping into it. You can ask AI what environmental DNA metabarcoding is. It will give you a clean paragraph, maybe even a diagram, and that's powerful. AI is already inside labs, running simulations, analyzing sequencing data, and optimizing experiments. Algorithms now map ecosystems faster than field teams. Machine learning screens compounds before a human ever touches them.
The last 10 years was really about the science of AI, but the next 10 years is going to be the application science of AI.
But here's the line. AI can process data. It cannot build depth. It can generate answers. It cannot challenge your assumptions. And most workshops? Still teaching what was impressive 10 years ago. Repackaged slides, safe techniques, outdated comfort. If you're comfortable learning yesterday's science, stay online. But if you want to understand where the field is actually going, then you don't need convenience. You need proximity. Because tomorrow's scientist isn't just someone who runs experiments. It's someone who understands how biology, data, and computation collide. Streaming is intangible. Showing up makes it rare.
Genetic engineering could become our most potent tool for managing the planet. The wonders of the future may not be built with concrete or circuits, but with cells.
Okay, with that,
Okay, right. Yeah.
yes, sir.
Yes, Pablo, go ahead. Yeah.
Yes, with that, so we can move to the Q&A session. So our participants has asked several questions from the beginning of the webinar as well. So I'll start
Yeah.
from the very first
Pablo,
question.
let's do something
Yeah.
like that. Yeah, since
Yes, sir.
we have a lot of questions, I'll pick one or two questions from the chat and answer, right?
Yes, sir,
And
of
then
course,
they
that's
can
better.
direct their questions in our, maybe can we create Discord, WhatsApp? Yeah, later we can provide comprehensive answers. Yeah,
Of course, sir.
and yeah, as Pabodi mentioned, everybody, we have a game. You have only 24 hours to find the mutation, right? So this is an imaginary condition that we are in trouble if you do not find the mutation, guys. We will not be able to create a guide RNA and tackle the problem. So you have 24 hours to design guide RNA. So you have to help scientists to save the country from this bacterial attack, right? So it's a Bioloom Island team is ready to create a guide RNA and Cas system if you quickly find the mutation and post it. And again, as Pabodi mentioned, our internship program is also very important. Pabodi, we are announcing it as Sri Lanka's actually first structured synthetic biology related internship program. So anyone interested can join with us. So it spans it was 3 months initially, and then another 3 months you can extend. Later you can work with us as junior scientists to conceptualize these synthetic biology and genetic circuits with CRISPR integrated platforms, so on. Yeah, so we are very excited to welcome you guys if you are interested in joining with our team. So we have intelligently developed this program for you all to gain the maximum out of it, guys. Yeah, I about questions. So as I mentioned, what you can do is I'm ready to answer all of your questions in WhatsApp. You can direct them in WhatsApp or via even via emails as well. So I came across an interesting one or two question. So this question, since CRISPR focuses specifically on minimizing off targets, how does its contextual scoring differ from the off target scoring algorithm used in ChopChop? Can you explain briefly? Yeah, that's a very important question. Whoever asked, the Mishana, right? Okay, if I'm correct. Right, the ChopChop, of course. So this is actually the ChopChop's algorithm. Is what it does is that Chop basically look at the guide RNA's compatibility with target sites. It's a sequence comparison algorithm. It compares its sequence and your target, and it scans for nearby sequence and see whether there are any other PAM sequences, and also whether there are similar sequence nearby, but in the genome. But when it comes to the CRISPR-off algorithm, so this is an intelligent algorithm, not only comparing the guide RNA's compatibility with the target sequence, it's a much more broader biological context when it comes to the CRISPR-off tool. Ms. Shana, if I'm correct again, so what happens in the CRISPR-off tool is that it's not only scanning, it also scan the chromatin accessibility, whether the how chromatin tightly packed, and also cellular context. So it studies the actual biological scenario and predicts whether that editing can happen precisely at this specific location. Basically, if I simplify, the chop has a sequence comparison algorithm and see whether there are similar sequences as well. But when it comes to CRISPR-off tool, it analyzes a much more broader, realistic biological context, like chromatin accessibility, as I mentioned, and also so many other biological interference. Epigenetic aspect is also analyzed in CRISPR tool, and then predict the accuracy of editing with that particular guide RNA. And there's another question I saw on, uh, wait a second. Can we apply CRISPR technology to reduce or prevent inherited genetic diseases before birth? Right. This is what everybody wants to hear. Excited about whether we can correct mistakes, whether we can give superpowers, whether we can correct traits in embryonic level. Uh, the problem, I don't know what's your name, Puta, but the thing is that currently with the regulation aspects, embryonic gene editing can be done in the laboratories for experimental purposes, but you are not allowed to implant edited embryos in wombs. What happens if so that step is not allowed? So you can edit for experimental purposes only, but you are not allowed to implant them. And see, the designer baby aspects, those are all futuristic topics and they are in discussion. So how precisely these can be deployed, right? I know, so that's the real power, but a lot of restriction and limitation, which makes sense because CRISPR has off-target effects and so on. So many things to consider. So those are the limitation. There's one person in China, scientist, who edited what do you call this immune system related gene, HIV resistant gene was improved with CRISPR tool, and his entire career was like suspended, and the lab, he was banned from conducting scientific research. So that's because he edited embryos and implanted them, right? So those kind of things are not allowed currently. Uh, some more questions. How can we use CRISPR to detect organ function reduction via identification of biomarkers using non-invasive method, such as using urinary analysis as example, kidney function? Yes, you can also analyze protein biomarkers as well. There are strategies like Cas12, Cas13. They can bind with the guide RNA supported DNA, but what you can do it, you can utilize Cas protein to couple with antibodies and then to interact with these protein biomarkers, and then you can utilize this as a sentient system. Of course, protein detection, small protein molecules can also be detection. It's possible with uh, CRISPR platforms. You can explore, there are articles on that, how you can use the antibodies as adapters to an aptamer-based detection is about the DNA binding protein. There are strategies called uh, what you call the ribo switches, aptamer-based strategies, and toehold switches. So those are the keywords I can give you if you are interested in extending your CRISPR workflows towards sensing aspects, guys. Right. Yes, everybody, I hope that you got something out of this webinar and you are excited to utilize these technologies and learn what's actually ahead of timeline. So you're always welcome to reach out to us via our WhatsApp platform and our email via email. So we are always there as much as we can. We are here to facilitate your educational journey and for you all to try as scientists, young scientists in the field. Okay, everybody. Yeah. The questions we will list out and definitely I'll answer all your questions. Our team will work on a list of questions that you have posted in our chat box as well. Right. Okay. Thank you very much, guys.
Okay, so thank you very much to you for this valuable and insightful webinar session. And with that, all of the guys, actually there is a small clarification that the CRISPR game is something different. So the CRISPR 0R is the game, and also there is a separate assignment that you have to submit to take your certificate. So all those details will be provided through our WhatsApp community. So follow those guidelines and then you can find out what you have to exactly do for the game and also for the assignment. Okay, right. So with that, thank you all for being a part of today's CRISPR webinar, and your enthusiasm and engagement have made this session truly inspiring. A special thanks to Dr. Lalinka Herath for sharing his expertise and guiding us through the exciting world of CRISPR, for fundamental gene editing concepts to practical applications in biotechnology. We hope today's session has sparked ideas and motivated you to explore how CRISPR can be responsible applied in research, agriculture, and beyond. Remember, gene editing technologies like CRISPR are powerful tools that can shape the future of science. But with great power comes great responsibilities. Keep learning, stay curious, and continue innovating. We look forward to see you in our upcoming webinars. Thank you once again. Have a nice day.
Welcome to Bioloom Islands. Bioloom Islands is a learning space built for the next generation of biologists. A place where molecular biology, computation, and curiosity intersect. Biology is evolving faster than traditional education can adapt. We introduce the new era of human and environmental evolution, synthetic biology. Yet many learners never get the chance to use them meaningfully. Bioloom focuses on practical understanding. We turn complex tools into usable skills through hands-on workshops, guided exploration, and real scientific workflows. Today's workshop is one island in that larger journey.